Wanted to try to connect with you about your ACVR1 grant that you applied for here to the, I think this one was to the Cure Starts Now, but I wanted to ask you a question about that if I could get your information on that. The ACVR1, this was talked about a lot, not only in the Medical Advisory Council, but also through the construct of Connect.
Right.
And that's where this is primarily based, correct? Is this is basically an offshoot of Connect?
Yeah, so this is an attempt to generate a huge amount of data in a short period of time. And so we are linking various labs associated with Connect to run all these experiments in parallel and then feed that, hopefully, back through the clinical end of Connect into a trial.
Okay, and is this proper, and that's how it's written, is this effectively going to identify new pathways of drugs? Is that what you're planning on with the results of this?
So there's two things really. One is just to screen existing drugs across our models to find the best one to move very rapidly into trial. And then because we assume that a single drug will not be completely curative, we also want to think about the next generation of trials and how we would combine an ACVR1 inhibitor with another class of drugs to really see a survival benefit, which is what we all want.
Tamara?
So, Dr. Jones, it is with great pleasure that we are going to award you $498,716 for your Translating ACVR1 Mechanism grant.
I can't peek over the check.
That's astounding, and I was genuinely not expected. As you guys know, I put this in last year, and obviously it's been an incredible year and a half for us all. And it wasn't successful then, and I totally understand the difficulties everyone's had with fundraising and with trying to win grants from our side. So it's a massive surprise. It's just incredible, and it's going to make a huge difference to what we can do to really rapidly translate those findings directly into clinical trial and hopefully directly into patient benefit as well. I mean, that's amazing.
Well, and it was a grant that obviously you went ahead and applied last time. I think you clarified some of the aims with it, and the Medical Advisory Council was very excited about what you put in with this, and frankly is very excited about what it's going to mean. You have not only between the Cure Starts Now and the DIPG Collaborative and Reflections of Grace and everybody else, there's 66 organizations that are excited about this. So no pressure. We're expecting that cures cancer. And that you're able to we're able to use those results in the future through the larger trial system here. So
yeah, absolutely. It's such a pleasure to work with you guys. We've had a long history, as you know, and with my involvement now in Connect, it's just super exciting to be able to put these things together, to use these funds just to really translate this data as quickly as we can.
Oh, fantastic. Well, I know while I got you here, if I can, I also wanted to ask you a little bit about your co-culture disruptor screens with that. I know, Tamara, you had a question on that one as well.
Right.
What was yours there?
So in your summary, you indicate how the DMG cells are codependent with each other and the other DMG cells. And a key aspect is to disrupt this communication. Could you talk about that a little bit?
Yes, this is something we're really excited by because, often when we try to find drugs which will kill these cancer cells, we're using a very blunt tool and just trying to stop the way they grow, stop the way they divide, stop the way they move even as well. I think we have a new sort of angle on that, which is also to stop the way they communicate. They're not one single cell type. There are these sort of subtypes within an individual's tumor. They do talk to each other, they cooperate, and this really drives the way they can grow and develop through the brain. So we think we have a different way of treating them, which is to block the way they talk to each other. And so what we hope to do is to screen a bunch of drugs, find the most effective at blocking this communication, and then maybe combine that with some of these other drugs that we've already been trialing and already in the clinic in a way that we hope we may really, increase survival in a much better way and have a much more effective way of killing these cells.
Well, and that seems to be something that's almost a generational scenario. I know we've been talking about that in a couple of different symposiums with it. And so this seems to be the next permutation of that. Well, also with that, Dr. Jones, you have a notoriety here that we have never had. You actually have won 2 grants in one grant cycle.
Wow.
And you've gotten full funding for the $113,747 for your co-culture disruptor grant as well. I've never awarded 2 grants at once.
It's just absolutely incredible. Again, this means a lot because some of the original ideas that went into this grant, as you may remember, came from DIPG collaborative funding several years ago. And like you say, we've talked about this at various symposia. It's such a pleasure, such a thrill to be able to use these funds to develop these ideas further, on the basis of, the original grant funding you gave us all those years ago. So it just means a huge amount. And yeah, I'm genuinely flabbergasted.
Well, congratulations.
Congratulations. We, the families are just thrilled to have you for so long and just, have you just dedicated your life to making a difference. And from, representing them, it's very hard to convey how thankful we are for people like you.
Thank you. That's incredibly kind. And just to reiterate that how thankful we are to you guys. We know the lengths that you go to support us, to raise the funds, to advocate on the researchers and the families' behalf. And it's such a pleasure to work in, collaboration and cooperation with you guys. And that's what makes this particular, funding body, this scheme, so extraordinary and such a pleasure to be a part of.